Okay. Hello everyone, how are you all doing? It's igcse.science.notes.z with Taqwa team, and here we will be doing, as promised, Unit 1 most repeated and most important questions and definitions. You will find this PDF on the Telegram channel of igcse.science.notes.z and the WhatsApp channel of Taqwa team. So let's get started with one of the most important questions that you will ever see. It might come in fill in the blanks, theoretical question, or even MCQ question. It says, explain why water is an effective molecule for transporting other molecules around living organisms. You get to say because water is a solvent and that water is slightly charged and that the polar molecules can dissolve in water. Water is viscous and that the water as a liquid assists mass flow. So that's why water is an effective molecule. Okay, because it's very important, let's repeat. Why water is an effective molecule for transporting other molecules around living organisms. Let's divide this part. Let's divide this thing. Why water is an effective molecule? Because it's a solvent and it's slightly charged and it's viscous and it's a liquid that assists mass flow. For transporting other molecules, because polar molecules can dissolve in water. Okay. Question number 2. Suggest two differences between the fibrinogen and the fibrin. I want you to know that fibrinogen is globular while fibrin is fibrous, and the fibrinogen is soluble while fibrin is insoluble. That's why in blood clotting and so the fibrinogen fibrinogen gets converted into fibrin, so it forms a mesh to stop the blood flow. Fibrin is insoluble and it's fibrous. There is that another question which is having the same thing that as we said right now. Describe the blood clotting process. So basically there is a cascade of events. The thromboplastin starts the cascade. Thromboplastin is an enzyme. The thromboplastin converts prothrombin into thrombin. This needs calcium ions. The thrombin converts fibrinogen into fibrin, as we said right now. So the mesh of fibrin forms and the platelets get trapped in the mesh. So again, the fibrinogen and fibrin is important in the term of blood clotting. The thromboplastin converts the prothrombin into thrombin, which needs calcium ions, so it converts the fibrinogen into fibrin. Why into fibrin? Why fibrin? Because it's insoluble. Okay. Question number 4 asks us to describe the structure of an artery and it's related to its function. Basically, the artery is very important to its function because it has elastic fibers, has smooth muscles, the muscles can contract, has smooth lining, reduces the friction, and has a narrow lumen. It also allows stretching to accommodate higher blood pressure and has folded endothelium. Question number 5. Describe the circulation of blood in a fish. So basically, we know from the IGCSEs that the fish has a single circulation, right? So the single circulation, basically, the blood flows from heart to gills. The blood flows from gills to the rest of the body. The blood flows from back to the heart. This is the single circulation. It doesn't pass onto the heart only once. Okay. Suggest advantage that the human circulatory system has compared with that of fish. You know that the human circulatory system is double, so passes to the heart twice. So why is that important or good? Has more advantage because the blood flows faster to the body, slower to the lungs, reduces the risk of damage to lungs with more efficient gas exchange system. Suggest why small organisms do not need a specialized gas exchange system. For example, fish, sometimes insects and also other bacteria, for example, doesn't even have a circulatory system because it has all from surface to cell inside the body. Insects and bacteria. Diffusion only is sufficient. They have low activity like bacteria. Explain why many animals need a heart and circulatory system. Because heart is needed to pump blood around the body, allowing mass flow, and many animals have small surface area to volume ratio, circulatory system is needed to overcome the limitations of diffusion. Many animals have a high metabolic rate. Question number 9 asks us to explain why multicellular animals require a respiratory system. Again, it has the same idea of the of the circulatory system, because diffusion over external surface is not enough, and that they have smaller surface area to volume ratio, and that the respiratory system has large surface area, so diffusion would be too slow. Thank you, Neer Chaudhary, for doing this PDF. Anyways, so just how circulatory system in mammals enables efficient gas exchange. Mass flow is generated by heart. Moving blood helps maintain a concentration gradient. A steep concentration gradient gives a fast rate of diffusion. Network of capillaries, large surface area increases the rate of diffusion, and the capillaries have very thin walls. Diffusion is fastest over small distances. No organs are far away from blood, and that the efficiency is related to double circulation. As we said again, this is very important. Why circulatory system in mammals enable efficient gas exchange? Because it has capillaries. Its capillaries have very thin walls, and it has network of capillaries. Also large surface area, which increases the rate of diffusion. This is important because the blood maintains concentration gradient and it's generated by the heart. This concentration gives a fast rate of diffusion. Diffusion is fastest over small distances. No organs are far away from blood. Efficiency is related to double circulation. Question number 11. Describe how the structure of the human lung is adapted for efficient gas exchange. Basically, it has many small alveoli covered by extensive network of capillaries, ensuring the large surface area for gas exchange, and that the thin capillary walls also increases the diffusion. Okay, second question. Describe the roles of the heart valves in the cardiac cycle. The atria contract, which is basically by the atrial systole as well. The atrioventricular valves open so that blood flows from atria to ventricles. Ventricles contract so that the ventricle systole occurs. The semilunar valves open so that blood leaves ventricles. The ventricles relax. The ventricular diastole. The semilunar valves close to prevent backflow of blood to ventricles. Okay. Question number 13 asks us to describe the role of the atrioventricular valves during the cardiac cycle. Guys, please, if you have any questions, I'm having the comments open right now. Ask me whatever you want. Where is the comments? If you ask, I'm here, active, okay? Describe the role of the atrioventricular valves during the cardiac cycle. Basically, the valves separate the atria from the ventricles, open during atrial systole, so that blood can pass through the ventricles, close during ventricle systole to prevent blood being forced back, which is the backflow, open during diastole so that ventricles can start to fill up. Question number 14 asks us to suggest why a faulty atrioventricular valve could lead to symptoms of breathlessness and lack of energy. Because the valves do not shut properly, and so that some of backflow of blood from ventricles to atria. So there is low blood pressure, and so less efficient supply of oxygen. So blood pressure in lungs increases. Basically, when there's backflow of blood from ventricles to atrial during the ventricle systole. Okay. Describe the structure of a mammalian heart. It has 4 chambers. The walls are muscular in nature. The heart is made of cardiac muscle. The ventricle walls are thicker than that of atria. The atrioventricular valves are between the atria and the ventricles. The semilunar valves are at the base of the aorta. The atria are on the upper side of the atria are on the upper side and the ventricles on the lower. Septum separates the right and the left side. Just guys, a small note, Unit 2 questions were covered earlier. We're doing Unit 1, and inshallah we will be doing Unit 4 and 5 as well. Unit 3 and Unit 6 are already on the channel. IGCS Edo Science Notes Z, right? So it's very easy, but there are multiple of PDFs if you would like to study by yourself from the PDF. It's also on the both channels right now, inshallah. I want you just to make sure that you're listening to what I'm saying, because in this question, it's going to be very important, guys. This is one of the most important questions. It asks us to describe how the atherosclerosis develops. Basically, it has first there is damage to the endothelial cells of the artery. So this causes an inflammatory response. The migration of white blood cells to that area occurs. So they build up cholesterol, formation of atheroma, narrowing of lumen. Process is self-perpetuating. Okay. Describe the location of the following structures and explain their functions. Semilunar valves is in the base of the aorta and prevents backflow of blood during atrial systole. Elastic fibers is in the middle layer of the wall of the vessel and it allows stretching to prevent damage to aorta. Explain the difference in thickness of the wall of the right atrium and the wall of the right ventricle. The right atrium has less muscle. Thickness is related to blood pressure required. Right atrium pumps blood to right ventricle. Right ventricle pumps blood to lungs. During the cardiac cycle, the changes in pressure that occur in the left atrium and the left ventricle are different. Why is that? Because the pressure changes are smaller in the atrium than ventricle, and so the atrium has less cardiac muscle than the ventricle. The atrium does not have to push the blood as far as the ventricle has to. The increase in pressure begins in the atrium before the ventricle. The atrial systole has to happen before the ventricle system in order for the ventricle to fill with blood. The increase in arterial pressure causes increase in pressure in ventricle. I want you also to know that we need to explain why the heart is divided into right side and left side, basically to keep the oxygenated and deoxygenated blood separated. This results in as much oxygen as possible being carried to tissues. Different pressures at each side of the heart occurs. Okay, give reasons why the phenia is a suitable organism for investigating the effect of chemicals on heart rate. Because heart can be seen, and the phenia are simple organisms, fewer ethical concerns because it is an invertebrate. Also abundant, they can absorb chemicals from the surrounding solution quickly. Describe the structure of storage. Describe the structure of the storage, and the glycogen and cellulose comes both in Unit 1 and Unit 2. And Unit 3 as well, I believe. Describe the structure of storage. It's made of alpha glucose, joined by condensation, forming glycosidic bonds. It's made of amylose and amylopectin. Amylose has 1,4 glycosidic bonds, and it's spiraled, while amylopectin has both 1,4 and 1,6 glycosidic bonds. Having 1,6 glycosidic bonds make an amylopectin molecule branched, and it's also starch is a compact molecule. Describe the structure of glycogen and explain why it's a suitable molecule for storing energy. Because it consists of alpha glucose, which is which is joined by 1,4 and 1,6 glycosidic bonds. It's branched structure, compact structure. But why is it soluble? Because it's easily hydrolyzed, leading to more glucose in smaller space, low solubility, does not diffuse out of cells, and no osmotic effect. Suggest the differences between a monosaccharide and a polysaccharide. Monosaccharide is soluble, small, sweet, no glycosidic bonds, osmotic effect, single unit, cannot be hydrolyzed, while the polysaccharide is totally the opposite. It's insoluble, it's large, it's not sweet, it has glycosidic bonds, no osmotic effect, many units, and can be hydrolyzed. Did you ask yourself why it has osmotic effect, glycosidic bonds, and many units can be hydrolyzed? Because they all have the same thing. Without osmotic effect. So yeah. Since it's a polysaccharide, it's made of many molecules joined by glycosidic bonds. That's why it has present. And since it's polysaccharide, it has many units, and so it can be hydrolyzed since it's already made by condensation reaction, so it can have hydrolysis reaction as well. Okay. Describe the structure of an amino acid. It contains one group and one amino group and one carboxyl group. It has central carbon atom. This binds with all groups. Describe the structure of a fibrous protein. Long amino acid sequence repetitive, no tertiary structure, chains lie parallel, joined by disulfide bridges. Explain why the combination of a high-fat diet and low activity levels may lead to CVD. Because there is energy imbalance, individual becoming overweight, increased blood pressure, obesity leads to diabetes, increased blood cholesterol level, damage to endothelium, formation of atheroma, and narrowing of lumen. Explain why different enzymes are involved in the formation of different carbohydrates. Because enzymes are specific due to shape of active site, only allowing certain substrate to bind. Question 29 is one of the most important questions as well in almost many units. suggest how mutation causes the structure to be altered. Basically, the mutation changes the sequence of base in DNA. The sequence of base is the primary structure. The primary structure is altered, different amino acid is made, resulting in different bonding between the R groups, changing the shape of the protein. Describe the three-dimensional tertiary structure of an enzyme. The globular, the active site, specific shape of active site, bonds between the R groups such as the hydrogen bonds. Explain why the primary structure of an enzyme determines its three-dimensional structure and properties. Because the primary structure determines the positioning of the bonds, the polar on the outside of enzymes and non-polar on the inside. Globular and soluble as well, determines the shape of the active site. Enzyme substrate complexes forms. Enzymes are specific. Almost last question of enzymes. Describe an experiment that could be carried out to investigate the effect of enzyme concentration on initial rate of reaction. Range of enzyme concentration at least 5, substrate concentration not limiting. Measure the volume of oxygen involved in every 20 seconds. Plot graph and calculate the rate from linear part of graph. Control variable pH, temperature, volume, concentration of substrate. Repeat at each enzyme concentration. Do not control experiment for comparison. Describe the process of DNA replication. The process is semi-conservative. The DNA molecule unwinds. The DNA polymerase lines up mononucleotides. According to the base pairing rule, hydrogen bonds form between bases. Phosphodiester bonds form between adjacent mononucleotides. And by condensation reaction. Describe the process of transcription in protein synthesis. The DNA molecule unzips. The RNA polymerase lines up RNA mononucleotides against one strand. The complementary base pairing between DNA and mononucleotides. Phosphodiester bond forms by condensation reaction. mRNA detached from DNA. Guys, if you don't really like studying from PDFs, which I feel like try your best to revise at least from, there are also handwritten notes on the IGCSE on the account, on the Instagram account. It carries the same name, guys, okay? IGCSE.science.notes.z. It's handwritten notes, but make sure to also revise from these notes. They are not really long, they're just short, around 10 pages or something. Okay? So I want you to know also how to describe the process of protein synthesis that occurs in the cytoplasm. This is basically the translation. It might come by this name or by coming by describe the translation. You need to know that the ribosome attached to mRNA, the tRNA carries an amino acid, anticodon-codon interaction between tRNA and mRNA, formation of hydrogen bond between tRNA and mRNA, peptide bonds formed between tRNA and mRNA, peptide bonds formed by condensation reaction, tRNA released from mRNA, ribosome detached from sequence on mRNA. Again, how translation occurs: ribosome attached to mRNA, tRNA carries an amino acid, anticodon-codon interaction between tRNA and mRNA, formation of hydrogen bond between tRNA and mRNA, peptide bonds formed between tRNA and mRNA, peptide bonds formed by condensation reaction, tRNA released from mRNA, ribosomes detached from sequence on mRNA. Describe the role of the following in protein synthesis. The messenger RNA. It's important, basically messenger RNA and RNA, right? So it's important because mRNA is a copy of the genetic code of the protein synthesized. It moves out of the nucleus and acts as a substance for translation. The transfer tRNA translation bind to an amino acid specific to amino acid, hold the amino acid in place. Basically, in RNA and tRNA, the tRNA is folded, mRNA is straight. tRNA has hydrogen bond and has fixed size, while mRNA does not have fixed size. tRNA has codons, mRNA has anticodons. tRNA has an amino acid binding site. Describe the differences between replication and transcription. Replication uses DNA nucleotides, uses DNA polymerase, referring to semi-conservative, copies both DNA strands, makes DNA double helix. Transcription uses DNA nucleotides, does not use DNA polymerase, uses RNA polymerase, not semi-conservative, copies only one strand template, and makes single strand mRNA. Okay, so I want you now to know, let's go by describe the structure of cell membrane. It has phospholipid bilayer, and we need to have a correct orientation and structure of the phospholipid in the bilayer. Also, we need to know why the phospholipids are oriented this way, the proteins in the membrane, two different locations of protein, extrinsic, intrinsic, and transmembrane membrane, the glycoproteins, and the cholesterol within the temperature. You might be sleepy by now, but it's very important to finish this revision because it's like most important questions, Only 9 pages. I hope you all score A stars. So let's go by the last 3 questions, starting with the last one. Describe the role of proteins in the active transport mechanism. The molecules bind to protein carriers. The protein carriers change shape. The molecules move against the concentration gradient. This requires ATP. The facilitated diffusion channels protein channel can open or close for large molecules to pass through membrane. Molecules move down a concentration gradient. Describe an experiment to investigate the effect of temperature on the permeability of cell membrane. Collect beetroot from the same place. Use beetroot of the same size. Wash beetroot thoroughly. Use water bath to change temperature. Repeat at each temperature. Calibrate colorimeter to 0 absorbance for water and set to blue-green filter. Last question, which is very important. We said that we need to know correct orientation and structure and explanation of phospholipids, right? So explain how the phospholipid molecules form a bilayer. Fatty acids are hydrophobic, and so they aggregate together. The phosphate groups are hydrophilic, and so associate with water. 2 monolayers form a bilayer. Thank you guys for watching this video. I hope you find it useful. I hope you all get A stars and achieve the grades you are looking for. Ramadan Kareem, everyone. Good luck and bye bye.